Hey everyone, and welcome to This Explainer. I am absolutely thrilled to have you here today because, honestly, we are looking at something truly mind-blowing. You know, when we think about evolution, we almost always picture physical, outward traits, right? Things like razor-sharp teeth, perfect camouflage, or massive rings. But today, we're going to explore a high-stakes evolutionary race for survival that happened entirely inside the body. We're going to dive deep into the hidden, microscopic wiring of our nervous system and discover exactly how natural selection literally forged the ultimate biological machinery for speed.
So, think about this for a second. How does your brain actually tell your foot to pull away from a sharp rock or a shockingly hot piece of glass in mere milliseconds? Seriously, take a moment to really appreciate that speed. It's a lightning-fast reflex, and it's happening inside you right now. But it doesn't just happen by magic. It requires a tangible, physical signal traveling through your body at incredible, almost unfathomable velocities.
Okay, so here is our roadmap for today's explainer. We've got 1, the need for speed; 2, firing the action potential; 3, the squid's giant solution; 4, the vertebrate insulation upgrade; and finally, 5, when the signals misfire. Let's just jump right in.
Section 1. The need for speed. In the relentless environment of the animal kingdom, whether you're the apex predator or the vulnerable prey, your survival very often comes down to just fractions of a second.
Animals absolutely must react quickly to survive sudden danger or, to seize fleeting opportunities. A slow biological signal simply isn't fast enough for complex survival. Imagine a snail trying to hunt a fish. If the snail's nervous system cannot process the scent of the fish, send that data to its primitive brain, and trigger a muscular strike before the fish simply swims away, well, the snail starves. It's literally life or death. Biology had a massive existential problem to solve here. How do you transmit complex information from a sensor, like a nose or an eye, to a processing center and then out to a muscle instantaneously?
That brings us to Section 2, firing the action potential. To find out exactly how biology solved this life or death speed problem, we kind of have to zoom way into the cellular level. We need to look at the microscopic machinery that creates the initial electrical spark in our bodies.
That crucial spark is called an action potential. Basically, it's a massive change in the electrical voltage across the membrane of a nerve cell, or neuron. But here is the truly fascinating part. An action potential isn't just some static spark that fades out over a distance. It actively regenerates. Think of it exactly like a perfectly spaced row of falling dominoes. Once you tip the very first one over, it knocks down the next and the next, completely rebuilding its momentum and carrying a full-strength signal all the way down the long extension of the neuron, which we call the axon.
Okay, let's dive into this microscopic domino effect, because the mechanics here are beautifully precise. Neurons have a specialized, highly sensitive gates in their membranes called voltage-gated ion channels. So in step 1, the rising phase, a stimulus hits the neuron and causes the sodium channels to suddenly pop open. Sodium ions, which carry a positive charge, just come rushing into the cell. This influx makes the inside of the cell highly positive. It's a rapid positive feedback loop. Really, the more sodium rushes in, the more surrounding channels open up. But, well, it has to stop eventually, right? So in step 2, the falling phase, a built-in inactivation loop actually swings shut to physically block those sodium channels. And at the exact same time, a completely different set of doors, the potassium channels, open up. Positively charged potassium ions rush out of the cell, forcefully bringing the voltage back down. Finally, in step 3, the undershoot, the channels reset themselves back to their precise resting state, locked and ready to fire again. This sequential, perfectly timed dance, sodium rushing in and then potassium rushing out, is literally the fundamental heartbeat of your entire nervous system.
All right, Section 3, the squid's giant solution. So, we now have our electrical signal. But as nervous systems evolved and animals became physically larger, sending that signal quickly over long, sprawling distances became a brand new, serious challenge. Natural selection had to find a way to speed up this action potential.
Notice how invertebrates, like squids and lobsters, actually solved this problem. They used evolutionary brute force. Natural selection shaped their axon structures for faster signaling by simply making the axons wider. It's basic physics. Think of a wide plumbing hose. A wider hose offers significantly less physical resistance to flowing water than a narrow, constricted one. In the exact same way, a noticeably wider axon provides far less internal resistance to the electrical current of an action potential, which allows the signal to surge forward much faster.
And some of these invertebrates took this basic physics principle to the absolute extreme. The giant axon of a squid can be up to a full 1 millimeter wide. To put that in perspective, that's about the thickness of a standard credit card, which, in the microscopic, invisible world of cellular biology, is absolutely massive. I mean, you can practically see a single cell with the naked eye. This giant, wide biological highway allows for incredibly rapid behavioral responses, giving the squid the lightning-fast muscle contractions it desperately needs to jet away from an incoming predator.
Moving right along to Section 4, the vertebrate insulation upgrade. Let's see how this builds into our own evolutionary tree. Vertebrates, like mammals, birds, and us humans, face the exact same fundamental problem of needing rapid signaling for survival. But we couldn't just rely on 1 millimeter wide axons. Our complex brains have billions upon billions of neurons, and if they were all the thickness of a credit card, there's no way they'd fit inside our skulls. We needed a completely different, infinitely more elegant biological engineering method.
And this brilliantly illustrates the divergence in evolutionary paths. On one side, we've got the squid's giant axon relying purely on brute force width. On the other side, we have the vertebrate axon. Vertebrate axons remain incredibly narrow, saving vital space, but they achieve astonishingly high-speed conduction through a revolutionary upgrade, highly specialized localized electrical insulation.
This revolutionary insulation is called the myelin sheath. It's meticulously produced by supportive cells called glia that wrap themselves tightly around the axon in multiple thick layers of mostly lipid membrane. It is completely analogous to the thick plastic insulation that encases the copper electrical wires hidden in your house to make them vastly more efficient and safe. Because of this thick myelin insulation, the electrical signal simply cannot leak out into the surrounding tissue. Instead, the action potential gets generated only at specific, uninsulated microscopic gaps called the nodes of Ranvier. So the electrical signal literally leaps from node to node down the length of the axon. This jumping mechanism is called saltatory conduction. I really want you to remember that term, saltatory conduction, because it is incredibly fast. That time-consuming, repetitive process of opening and closing all those sodium and potassium channels we discussed earlier, it only has to happen at the tiny gaps, rather than along the entire continuous length of the nerve.
The evolutionary contrast here is just incredibly stark when you look at it. The squid giant axon is a massive 1 millimeter in diameter, has absolutely no insulation, and is forced to use slow, continuous conduction. The vertebrate axon, however, is a tiny, practically invisible 20 micrometers in diameter. By adding that revolutionary myelin sheath, the vertebrate axon completely unlocks that saltatory or jumping conduction.
So the crucial point here is the major selective advantage this provides: unbelievable space efficiency. Because of that brilliant myelin insulation, a vertebrate axon that is a mere 20 micrometers wide actually conducts signals significantly faster than the massive squid giant axon. This means you can comfortably pack more than 2,000 highly insulated, lightning-fast vertebrate axons into the exact same physical space occupied by just one single squid giant axon. It's an absolute masterpiece of evolutionary engineering. Honestly, it's what makes human consciousness and complex thought physically possible.
Which brings us to Section 5, when the signals misfire. Because as with any highly intricate, finely tuned biological machinery, things can occasionally go wrong. We really need to ground all this fascinating microscopic science by shifting to a more serious perspective, looking at what actually happens when these delicate, high-speed systems malfunction and mutate in real human beings.
Remember those tiny voltage-gated sodium and potassium channels we talked about operating during the action potential? Well, spontaneous mutations in the specific genes that encode those critical channel proteins can have profound, life-altering medical consequences. Depending entirely on where in the body the newly mutated channels are physically located, it can severely alter normal nerve, brain, or muscle function. It often causes localized groups of cells to fire excessively or simultaneously when they should be resting.
A painfully clear example of this is epilepsy. Certain forms of epilepsy can be directly caused by tiny mutations affecting the sodium channels right inside the brain. Remember the falling phase of the action potential, where the open sodium channels are supposed to be securely blocked by a swinging inactivation loop so the nerve can properly reset? Well, if a genetic mutation prevents those sodium channels from closing properly, that powerful positive feedback loop runs completely out of control. Massive groups of nerve cells fire simultaneously and excessively, which produces intense seizures. This isn't just abstract biological theory. It's the physical reality of how a tiny cellular gate failing to close cascades into a massive disruptive neurological event.
Now, if we take that exact same type of microscopic malfunction, a mutated, faulty voltage-gated sodium channel, but place it in skeletal muscle cells instead of the brain, the real-world result is completely different. It leads directly to a debilitating condition called myotonia. This causes a periodic, uncontrolled spasming and stiffening of the muscles, because the delicate electrical signals commanding the muscles to contract and relax are completely misfiring at the deepest cellular level. Everything we've explored together today, from the lightning-fast opening of a single microscopic sodium channel to the massive, protective insulating wraps of the myelin sheath, all comes back to this fundamental, undeniable truth from our source material. The rate at which axons conduct action potentials governs how rapidly an animal can react to danger or opportunity. Evolution didn't just build us to passively exist in the world, it meticulously wired us for speed, unmatched efficiency, and ultimate survival.
Which leaves us with a truly fascinating, open-ended thought to end on today. We've seen exactly how billions of years of ruthless trial and error took us from a slow, continuous signal in the ocean to a lightning-fast, highly insulated, jumping action potential in the human mind. If a single microscopic ion channel mutation today can so profoundly alter human consciousness and physical movement, what incredible hidden evolutionary upgrades might our nervous systems undergo next? Thank you so much for exploring the intricate, beautiful wiring of life with me in this explainer. Keep wondering, keep learning, and I'll catch you next time.